Hi everyone. My name is Francis Dinh. I'm an undergrad at McGill University, so not too far from here. And before I even start this presentation, I just want to go into the acknowledgements real fast. I want to thank all my collaborators, Isabella, who gave me the GIS data that I needed to do my analysis, and my co-supervisors, Allison and Andrew, who just guided me through this whole process. It is really an honor to get to talk at Evolution today. So let's get into it. So I guess the general framework that I want to understand is parallelism in urban environments. So we often think of cities as being these sort of environments where there's lots of convergent features, and I guess we can use these convergent features to try to understand convergent evolution across different cities. But the question always remains in evolutionary biology is how convergent are these declines? And as we know, any kind of environmental variation can lead to very different landscapes very different adaptive landscapes in the species that we're studying. And there's also always the effect of other processes like genetic drift or founder effect or gene flow that can affect the just urban evolution in general. So I'm just going to go and start talking about my model species, which is the eastern gray squirrel. For anyone living in the east coast of North America, these gray squirrels don't even need an introduction. They are ridiculously common all over urban areas. They like to feed off of donut holes at campus. They're really all over the place, and I guess that's why we like to use them to understand urban ecology. So the eastern gray squirrel is typically gray, but there's a good amount of them that are melanic, and this melanic morph results from a deletion at the MC1R gene that is dominant, and it causes them to be either pitch black or brown, dark brown. So it's a pretty straightforward dimorphism that exists in this species. The other thing that needs to be noted about the melanic morph is that they used to be a lot more present in North America, but their prevalence sort of waned out over the past 200 years. But they do maintain some pretty strong prevalence in northern, more cold areas of North America, where it is believed that maybe this morph has a better thermal regulation, and it also maintains a bit of prevalence near cities. And that is the question that Squirrel Mapper wants to understand. What causes the trends in distribution of urban gray squirrels? So Squirrel Mapper is a citizen science project that collected a bunch of iNaturalist data from urban squirrels, and what they've noticed is that we do typically see an increase of melanism the closer we get to big cities in North America. Specifically, this effect is particularly strong in large cities that have extensive forest cover and which have cold winter temperatures. And and we can look at the data here that was published by Costantino and Gibbs in 2022. It's a great paper, and we can see how all of these cities up here have this nice trend of increasing melanism with the higher urbanization. The problem, though, and the one that I'm trying to understand is why for maybe the majority of cities, we don't really see that much of an increase in melanism with higher urbanization. In Montreal, there is basically no relation between melanism and urbanization. So that is the question. Is there any spatial structuring in this city in particular? So let's start with the general Montreal area. Montreal is situated on an island, but this island is separated from the rest of the metropolitan area by the St. Lawrence River. And there are several different regions, for example, Laval Island here, that is separated by this really tiny river, and there's other places like the North Shore here that is particularly rural. There's a South Shore that is actually situated mostly east of the island, interestingly, and there's the west shore down here. So what I did is that when I had time off of my part-time job in summer 2022, I took my bicycle and rode it all around to the Montreal island, visited 200 different parks, and conducted visual surveys of the population that lived there. And just to talk real quickly about what I saw, first off, squirrels were present in 152 of those parks, which really brings back the point that they're like omnipresent in the city. And melanics were present in 46 of those parks. So they're relatively all over the place. But what seems to be more interesting is that there is like a really high variation in melanism across different areas. So for example, on Montreal Island alone, which I have to like mention, is often separated in three parts: the West Island, the Central Island, and the East Island. The West Island is often like more separated from the other areas because there's like a lot of like transportation hubs there, and it creates like this sort of like separate area that I am using in my analysis. So if we consider all these different areas in the Montreal area, we see that different regions just have such a great variety and just such a great divergence in their proportion of melanism that I'm not sure how to explain it otherwise than just the fact that regionalism is a big predictor of the distribution of squirrels. For example, look at the island, like the central areas of Montreal, which have somewhere between 10 and 16% of the population being melanic. Compare that to 40% of the population in the North Shore, whereas in Laval Island, which is right in the middle of the two, I personally did not see like one melanic squirrel there. I'm not saying it's not there, but it is at least very rare, very, very rare. Even in East Island here, which as I mentioned, is often thought to be sort of separate from the rest of the island, there's maybe somewhere between like 15 to 20% of the population being melanic. So that is that is pretty interesting, I think. The spatial autocorrelation is pretty weak. It is significant, but like I think regionalism is more important here. And if we use distance from city center as a proxy for urbanization, we actually see that in Montreal, there is overall an increase in melanism as we get further from the city. But if we look at specific areas, for example, only looking at the island of Montreal, this effect is a lot weaker. Maybe possibly due to gene flow, who knows. And in some areas, like the South Shore, that is basically we basically see the opposite trend. And yeah, if we look at how different these ratios are, well, the North Shore has a ton of black squirrels, lots of them in the west, a few of them in the center and in the east parts of the island, which from now on I will consider as sort of the same place. There is a good number of them, but in Laval and in the south, there's really not that much prevalence. And just to make sure that this is not just another one of these rural urban trends, for example, maybe like the central part of the area is just like more urbanized than everything else, right? Well, these different areas don't really seem to have that much like difference in terms of urbanization, which I measured as the building area around the sites that I sampled. So compare that with the slide before, and we don't really see like that strong of an urban-rural trend. So if we look at divergences between other environmental factors, such as like canopy cover, like the amount of, like how much of the park is covered in trees, once again we see that overall there is a pretty solid increase in melanism where there is more trees, but in Montreal that effect is a lot weaker, and in the South Shore where they're basically like where there's a lot less of them, there is like not that strong of a relation. Lastly, let's look at the areas where where they do have prevalence, where they do have presence, and we start to see that even a simple relation, such as how much of the squirrel how much does the squirrel population correlate with higher tree covers, we see a pretty solid divergence in trends in different regions. For example, on the North Shore, they just really solidly increase with higher tree cover, and they sort of increase, but not to the same extent on Montreal Island, and in the west, that trend is kind of reversed. So my point being that there does seem to be a good relation between urbanization and the prevalence of melanism, but this effect is actually either weak or it is highly variable because of other factors. Notably, the large differences in the prevalence between different regions in my data set seems to suggest that the rivers may cause like significant barriers to movement, resulting in divergent populations, which results in divergence in environmental responses. So I think that maybe it is non-deterministic processes like genetic drift or gene flow or the founder effect that really are the strongest predictors of spatial variation of polymorphism. For example, maybe if we look at Laval Island, which had very, very little squirrels, maybe the first very, very little melanic squirrels, maybe the first one that got there was gray, and due to the founder effect, it just really took over the entire island, and that subsequently any attempt at colonization by like melanic squirrels just led to the allele just kind of like getting washed away over time. So I guess that's the end of my presentation. Thank you very much for listening. It is really, really an honor to be here. And here's a bit of my contact. You can email me if you have any more questions, or you can talk to me on Instagram. I like that. Yeah, and I'm applying for grad school this year, if anyone is looking for a prospective student. Thank you. Yes?
I really enjoyed your talk. I got to see, because I grew up in a place that had the black squirrels. They always wondered what was going on. I was interested to know how you might expand on this research if that's something you were interested in doing.
Yeah, well, I am interested in expanding the research because, well, part of it actually, this project came up actually as sort of like a little extension of a more established project of Squirrel Map. Oh, sorry. Is that 3 more minutes? Okay. As an extension of Squirrel Mapper, led by Gibbs and Costantino. Is Brad in the room? Hey, yeah. So, I think I would like to expand on other factors, maybe like predation within the Montreal area. For example, in other cities, it is believed that the relaxed selection in cities causes this urban-rural gradient. Well, perhaps in Montreal, which is a completely different geographic context, maybe there is a different adaptive climb that is going on here. Maybe in the north, there's like just a lot more forest, and that's where the natural, like the squirrels tend to live, and maybe that's why we see less of them closer to the city. And yeah, if there's any other features like structuring or predation that can be explored, I'd love to talk to anyone about it. Yes?
This might be me being a little bit naive, but living up in the UK, I've never seen a black version of a bird squirrel. Do you think them being invasive in Europe, there would be the same sort of patterns with the urbanization?
I haven't looked into patterns in Europe. Yeah, sorry, I don't know how to answer your question. Any other questions? Yeah?
Yeah, I was curious. It seems like there's also a latitudinal climb.
Yeah.
In addition to the urbanization rate.
Mm-hmm.
I was curious, is there much understood about what drives the latitudinal climb?
Yeah, so the latitudinal climb, what we believe is happening is that in colder areas, therefore like more northerly areas, the black morph is adaptive because it has better ability for thermogenesis. So in the south, where where temperatures are a lot hotter, like the black morph is almost absent. Thank you.
Okay, there we go. Hello, everyone. Good afternoon. Thanks for coming. I'm Malin Pinsky, Associate Professor at the University of California, Santa Cruz. We've got two wonderful co-authors on this talk, Brendan Reed, also at UC Santa Cruz, and Bastian Starr at University of Oslo. So I lead the Global Change Research Group, where we work to understand how species adapt and when they don't to rapidly changing environments. We use mathematical theory to help set expectations, analyze large ecological data sets across time and space to make sense of ecological adaptation. And the focus of today's talk, we use temporal genomics to understand evolutionary responses. So for many species, evolution is often thought to be too slow to be an important process for species coping with climate change, with novel hunting pressures, or with other novel environments. And yet it's also possible that our tools for detecting rapid evolution have also been relatively weak and not all that powerful. And as today's session, I think, is going to illustrate, there are a growing number of exciting examples of contemporary adaptive evolution. A key concept in this field has been the idea of evolutionary rescue. So in this figure here, population has just experienced a change in the environment. Absolute fitness is below 1, and the population's declined to extinction. But if the population can evolve to tolerate the novel environment, then it can recover, and that's the process of evolutionary rescue. So for these questions, the tools of temporal genomics provide a powerful window into those evolutionary dynamics. This is just the application of DNA sequencing to repeated samples from the same population through time, so we can observe evolution in action. Evolution, after all, at its core is a change in allele frequencies through time, and the dynamics of those alleles provide some indication about the processes that are driving that change, including positive and negative selection or neutral drift. So those genomic signatures, though, also depend on the architecture of the traits that are under selection. So monogenic traits are driven by a single locus of large effect. Think melanism in peppered moths favored by industrial development. And a lot of what we understand actually comes from these single loci of large effect because they're the easiest to detect. Oligogenic traits are driven by a few loci, polygenic by many loci, all the way up to omnigenic, where it's effectively the full genome that's contributing to the phenotype. So our story today is about fishing. And as actually we heard earlier in the symposium, the examples of contemporary phenotypic change, of those many examples, the most extreme all involve fishing. And that was actually something that J.B.S. Haldane predicted more than 90 years ago when he said that fishing could lead to extreme and abnormally fast evolution. And from a theoretical perspective, the mechanism seems pretty clear. Fishing increases the mortality rate, so individuals don't live as long. Those with alleles for reproduction at earlier age and smaller size are more likely to leave offspring in the next generation, driving evolution towards earlier reproduction. Those changes can also have substantial ecological and economic consequences, including lower population stability, greater vulnerability to predation, and also just smaller fisheries catches. So in the wild, many species of fish, and especially Atlantic cod, now mature earlier. And that decline in body size, in particular of Atlantic cod, has literally become a textbook example now of rapid contemporary evolution. All this happened during intensive fishing after World War II, around the same time that many of these populations collapsed to very low levels. The challenge, though, has been that this is also the phenotypic change you would expect from plasticity. With fewer competition from mates, you also expect individuals to be able to mate earlier and at smaller size. So this debate between evolution and plasticity is part of what led Bastian Starr and the archaeogenomics group to conduct whole genome sequencing from historical samples from 2 Atlantic cod populations. 1907 from northern Norway, about 20 individuals, and similar from 1940 in eastern Canada, plus contemporary populations in the same, contemporary samples in the same locations. So in both of these populations, the phenotype has changed from maturation about 7 to 8 years to now as little as about 5 years in more recent phenotypes. So the whole genome resequencing led to 8 to 10x coverage across these 113 individuals. We mapped reads to a whole gene, effectively a full chromosome genome assembly, and called probabilistic genotypes. So the first thing we saw was clear evidence that these samples were from the same populations through time. This is a principal components analysis, lumping the historical and modern Canadian samples together in yellow, and the same for Norway in orange. An admixture analysis where each individual is a vertical bar organized by geographic location of sampling, Canada and Norway, and by time from left to right. Again, clusters them into two continuous populations through time. So it does look like we're sampling from the same populations. Allele frequency changes through time are quite weak, about 1%, suggesting that effective population sizes were very large, like on the order of 10,000 in Norway, maybe 35,000 in Canada. That's from a Jordi Ryman estimator. And we didn't detect any genome-wide declines in genetic diversity. The main focus, though, was looking for evidence of selective sweeps and selection on particular locations in the genome. One expectation would be regions with elevated FST values. So that's on the vertical axis here for Canada on the top, Norway on the bottom. Across the linkage groups, so effectively the chromosomes in the cod genome across the x-axis. And none of these regions show statistically elevated FST. We applied a whole range of other methods to look for selection targeting particular regions of the genome, and none of them identified clear targets of selection through time. So that forced us to first consider that maybe this actually was an example of phenotypic plasticity, and that these changes actually are not an example of rapid contemporary evolution. Another possibility, though, is that it was evolution, but just polygenic, spread across many loci, each of which changed only a very small amount that was not detectable from the sample sizes that we had available in this case. So polygenic selection, though, does leave a signal in the genome, especially in replicate populations. You expect the same alleles to change in the same direction when they experienced similar selection through time. That appears as a positive convergence correlation statistic, and that was laid out as theory by Vince Buffalo and Graham Coop a few years ago. So most applications have been to lab populations where it's easy to sample through time, but we were excited to apply this in one of the first examples to populations in the wild. So this is work that was led by Brendan Reed, postdoc, now a research scientist in the Global Change Research Group, soon to be a research faculty at Rutgers University in New Jersey. So we could compare the change in Canada against the change in Norway as a test for polygenic selection using either the first or the second modern Norwegian sample. As a positive control, we could compare the change in Norway against itself, and as a negative control, compare the change in Canada against the sampling noise between the two modern Norwegian samples. So again, we've got the linkage groups across the x-axis. Larger convergence correlation statistics suggest more evidence for selection. The negative control shown here in orange was consistently near 0. The positive control consistently positive across the genome. And the two tests for polygenic selection were also consistently positive across the genome, shown there in green and in blue. We're also curious, though. So these are populations in the wild. We have to think about other factors like gene flow, probably low across the Atlantic, but still a possibility. There might also be background selection on shared deleterious variants in these two populations. So we did a whole suite of simulations using SLIM to test those mechanisms. But I have to say, neither process seems to explain the patterns that we observed in these Atlantic cod populations. So we've heard a lot about inversions at this conference today over the last few days. So we tested, and the cod genome, as we heard earlier in Katie Lauderhaus's talk, does have many inversions. So we also tested whether polygenic selection was focused on particular inside or outside of these inversions. It doesn't appear to be. If anything, it appears to be concentrated outside the inversions on linkage group 1 and 7, rather than inside. Doesn't seem to be concentrated either in coding regions or in non-coding regions. There's probably a lot more, there's clearly a lot more to be done here to look at different functional classes within the genome, to look at particular molecular pathways. But at least at the moment, it appears to be quite an omnigenic response across much of the genome. So in many ways, this is actually good news for cod. It suggests that the genetic basis for maturation at later age is still present within these populations, and that recovery to historical phenotypes is still possible. The theory for fisheries-induced evolution would suggest, though, that that recovery is likely to be very slow because selection is going to be quite weak in the other direction. I just want to point out, though, that in this orange time series for Norway, over the last 2 decades, there actually does seem to be some recovery towards a later age of maturation. This is at a time when the Norwegian population has grown quite a bit and fishing has been dramatically reduced. Whether this is plasticity or evolution, though, is going to be a question for another day, but at least a hopeful sign for these populations. As I end, I also want to mention that we're continuing similar research, not just on single species, but actually across dozens of coastal marine fishes, all collected from the same location in the early 1900s. So this was a collection made by the research vessel Albatross. Samples have been stored effectively in high-proof rum for the last century at the Smithsonian. And if you want to hear more about some of the results coming out of that, go listen to Renee Clark's talk. It starts actually in just 15 minutes in room 516B, Anthropocene Genetic Diversity Loss in the Marine Tropics. So just in summary, I showed genomic evidence that this rapid decline in age of maturation in heavily fished Atlantic cod was accompanied by and maybe driven by highly polygenic selection. So some of the first clear genomic evidence that this actually was evolution in Atlantic cod. More generally, I think it's a nice example of how temporal genomics can reveal evolutionary dynamics that otherwise would be effectively invisible to many of the other methods that we have. And I think as our methods for detecting contemporary evolution become more powerful, we're going to discover that even in some long-lived species, evolution is a much more important mechanism for species coping with global change than we've realized so far. So thanks so much for listening. Again, want to mention Brendan Reed and Bastian Starr, a set of wonderful collaborators, funders that make this all possible, the many members of the Global Change Research Group that really make this rewarding. And we do have a fantastic grad program at UC Santa Cruz, so if you're a student interested, please do find me or drop me an email. Thanks so much. I think I've got time for a few questions, or a couple. Yeah?
Given the low-pass nature of the data, do you think there's enough there to market in the UTR regions and photo sequence? I think, you prefer to breaking down because
Yeah.
all these rapid evolution, we often end up people in expression.
Yeah. So pairing it with expression, I think, would be a great way to go. And bringing this into a more of an experimental system. I think it's going to be tricky, being highly polygenic. So, I don't expect that we'll be able to nail it down to particular loci, since, after all, it is polygenic, but particular person.
Recounting statistics, slowly looking at the origins and seeing if things become a little clearer. Right?
Just the sorry.
UTRs.
Oh. No, I think that could be interesting. Exactly. That's what I was. Yeah.
Basically, the low pass was the number of sites per window, and as far as
Yeah.
how many genes, how many UTRs there, whether it's going to be a global signal or if you look at it from the background, it's going to be very fast.
No, exactly. That's exactly what I think we need to do next. So, yeah, we haven't done it, but yes, I agree. Yeah.
So, technical question about the convergent evolution signal we're
Yeah.
detecting. Is it in vitro genotypes or just a population level
I heard part of the question, but not the whole thing. So, we have allele frequencies from 2 time points in 2 populations.
Yes.
And that's what allows us to calculate that convergence correlation statistic.
Individual genotype?
Correct. We do not. Yeah, and we don't. We have probabilistic genotypes rather than hard genotype calls in this case. So we have allele frequencies, not individual genotypes. Yeah.
Hi, everybody. I'm Isabel Novick, and today I'm going to talk to you about the life history, evolution, and divergence of this really interesting global pest species, Tiniola bacelliella. So I just want to start off talking about synanthropy. Synanthropy is when an organism adapts to and benefits from living with humans. Some examples you might know are pigeons and raccoons, and if you are unlucky, the webbing clothes moth. And we care about synanthropy because synanthropes can host zoonotic infectious diseases. They are pests in our homes and gardens and crop production, and they can damage our possessions in our homes. And this is relevant to us because urbanization leads to an increase in synanthropy, and as we continue to urbanize, we will continue to encourage synanthropic adaptation in the organisms that are all around us. I'm interested in synanthropic organisms and what traits predispose them to successfully adapt to the anthropogenic environment. So that brings me to this really interesting family of moths called Tiniidae, which contains a lot of species of synanthropic pests. They're one of the oldest ditrician moth families, and Ditricia makes up about 98% of Lepidoptera, and since tiniids are such an old lineage and they're basal in the tree, knowing about their diet evolution is really important to understand how Lepidopteran diet may have evolved. There's 3,500 species and 320 genera, and they feed on really weird stuff. They feed on detritus and fungus, and most notably keratin. And keratin is the protein found in hair, skin, and nails. And that's really weird for a lepidopteran, like a butterfly or a moth, to eat keratin, which is like horns and skin. It's really unexpected and very unique. But the systematics are really understudied, so we have this huge knowledge gap about this family because they are really, really small. They're micromoths, and also they are so cryptic, and they're really hard to tell apart phenotypically. So a lot of them, these are all different species, and some of them are different genera, but they look really similar to one another. And that brings me to the focus of today's presentation, which is Tiniola bacilliella, or the web-enclosed moth. These moths are found all over the world. They are globally distributed, and they have a lot of fun adaptations that make them really robust pests, really difficult to get rid of. And some of these adaptations are they can produce their own water as a byproduct of keratin digestion. They can digest heavy metals like arsenic, lead, and mercury. And they can chew through and digest synthetic materials. So this, as you can imagine, makes them really difficult to deal with when they're like a pest in your home. So my aim of this study is to investigate the evolutionary origins and biogeography of this globally distributed pest. And my first objective is to reconstruct the species-level relationships within Tiniidae to determine how diet evolution facilitated synanthropy. And my second objective is to determine the genealogical relationships among the current Tiniola bacilliella populations. So let's start off with objective one. My hypothesis for this portion is that synanthropy will be associated with repeated independent evolutionary origins and a keratin-based diet, and that their history of dietary keratinophagy just primed them to feed on indoor stuff. And I'm hypothesizing this because there's so much synanthropic and dietary diversity in this family, it would just make sense that synanthropy evolved multiple times separately and would be associated with keratinophagy. So how I did this, reconstructing this phylogeny was challenging because of how old and deeply diverged this family is. So to do this, we used ultra-conserved elements, which are targeted enrichments of conserved genomic regions. So all the moths have them, and it's really phylogenetically informative based on the variable flanking regions on either side of the ortholog. And they're a good method for reconstructing a family like Tiniidae because they're good for non-model organisms, they're good for resolving deeper taxonomic relationships, and I use BEAST for divergence time estimation and the PhyTools R package for ancestral state estimation. So this is really exciting. It turns out that this method using UCEs worked really well, and we were able to fully resolve genus and species level relationships within Tiniidae in this maximum likelihood tree. This is a really well supported tree. I don't know if you can see it, but those are all hundreds, those bootstrap values, and confirms phylogenetic relationships. And it's really exciting because we previously didn't have any phylogenies based on nearly this large amount of loci. And here on this tree is Tiniola baseliella, and what we found is that Tiniola and Tinea are sister genera. The Monopus genus is monophyletic, and the Tinianae subfamily is paraphyletic, and so is the Tinea genus. And now that we have this robust phylogeny, we can look at it a little bit more closely. So I made this divergence time estimated tree in Beast, and it took me like 6 months, and here I'm gonna talk about it for like 20 seconds. But the main message is that the Tiniidae family is super old, really ancient, and diverged about 150 million years ago. And Tiniola diverged about 27 million years ago. And just like to put this into context a little bit in regards to humans, humans, as we are now, diverged from Neanderthals about 800,000 years ago. And humans started building dwellings about 500,000 years ago. And we, as humans, started wearing clothes about 170,000 years ago. So Tiniola baseliella has been around and eating keratin for much longer than this. So we may consider them pests, but they're just doing what they have evolved to do over the years. So finally, I did an ancestral state estimation to assess support for my hypothesis that synanthropy is associated with keratinophagy. So first, the species name colors are denoting synanthropy status, and the node colors and the pies denote estimated dietary strategy. And it appears that the ancestral dietary strategy for the Tinianae subfamily, at least, was most likely facultatively keratinophagous, and the majority of species in that subfamily are at least facultatively synanthropic. So it seems that facultative keratinophagy was an ancestral trait that has led to facultative and in some cases obligate synanthropy. So this supports my hypothesis that there are multiple evolutionary origins of facultative synanthropy, and they are clearly associated with a facultatively keratinophagous dietary strategy. So on to the second objective. We can clearly use that phylogeny for a lot of really interesting things, but the second objective is to determine the genealogical relationships among the current web-enclosed moth populations. And this species contributes to like billions of dollars of damage per year in the US and has those really interesting adaptations I was talking about. But they are obligately synanthropic, and they're no longer found in non-human habitats. They're facultatively keratinophagous, so they are kind of opportunistic, and they're really poor flyers, so they have a relatively poor dispersal ability. So because of all of these factors, I hypothesize that there will be significant population structure between disparate populations of T. baseliella due to reproductive isolation and poor flight ability. And even though they're really broadly distributed with humans, they might have significant population structure. And I'm curious if that population structure exists, like, does it exist at a continental level, or are there more local determinants? So for this objective, I sent moth traps all around the world, and I used 2BRAD for sequencing, which is a reduced representation popgen approach. And this is good for intraspecific relationships, and is different from UCEs in that it captures more variation around the genome rather than focusing on specific orthologs. And then I used angst to call SNPs based on genotype likelihoods, and I did some PCAs that I will share with you. But first, just to talk about where they came from, there was a total of 288 moths that I used. Most were from America, North America, Europe, and Australia, but some were also from the Middle East, South America, and Africa. And you might notice that like the entire continent of Asia is missing, but that is not for lack of trying. I sent a bunch of traps out, and they came back with a different species. So it seems like there is a different dominant species in across Asia, which is pretty interesting. So here's a principal component analysis showing population structure for the global distribution of moths. And PCA is a way to visualize genetic relatedness. So samples that are closer together are more related, and the further away they are, they're less related, and each point represents one moth. So looking at this, we can see that there is some divergence based on geography, like looking at the UK and Germany. But there also appears to be divergence driven by unique locations within countries. So for instance, if we look at Austria in the blue, these two groups from Austria were collected from two different museums. And the museums were only 2.4 kilometers apart, but there is still a large amount of divergence between these samples. So there's a pattern of small-scale population structure, which brings me to my next PCA, which was all Massachusetts samples. I'm at Boston University, so there you go. And you can just use your imaginations that there are points inside of the ellipses. Interesting. Okay, so for this analysis, I reran angst on only the Boston area samples, and I did a PCA. And we can see clustering based on small-scale distance here. So Waltham and Cambridge are clustering separately, but it just doesn't seem to be based on distance. These towns are not far away from each other at all. They're all less than 20 kilometers or so apart, and the overlapping towns vary in distance as well. They're not any closer together than the genetically divergent towns are from each other. But the FST between them is still really, really high between the towns. And this is really interesting and indicates that there might be more cryptic diversity than we thought, or there's maybe significant inbreeding within populations leading to an inflated FST value. So we're gonna have to collect more Boston area samples to really tease this apart. And these PCAs support the hypothesis that there will be population structure, and yes, we can reject panmixia. However, absolute distance is not the best predictor of structure. Even within countries on very small spatial scales, there is structure, for instance, in Austria or within these Boston area samples. So to summarize, I'd just like to remind you that my hypotheses were that synanthropy would be associated with keratinophagy and multiple evolutionary origins, and that there would be significant global population structure. We have this well-resolved phylogeny for Tineidae, and it indicates that facultative keratinophagy is linked to synanthropy, and there are independent evolutionary origins of synanthropy. And there's also evidence of substantial population structure. However, it's not really clear yet what factors are shaping these patterns of genetic structure. Preliminary evidence suggests that local urbanization are barriers to gene flow, and urbanization is a really important factor. However, we still need to do some work on this to look at these patterns. All right, thank you, and I will take any questions that you have. Yeah.
I was wondering, versus time estimation, did you use fossils at all, or what kind of?
Yeah, I wanted to use a fossil, but there's only one fossilized Tineidae in amber, and we don't know what kind of Tineidae it was. It was just described as Tineidae by like Grimaldi, 1997. And so instead, I used secondary calibrations from the Kawahara 2019 tree. So I don't have any actual fossils calibrating it, but I have the secondary cals. Yeah. Yeah.
Yeah, so, first off, great talk.
Thank you.
And you were saying that the wasn't really determined by, like, actual distance. Do you know, is it possibly related to the amount of human traffic between those areas?
Could you repeat that? I'm sorry, I couldn't fully hear it.
So I was just curious about, like, the FST, like, not being driven by, like, absolute distance. Is it instead driven by the amount of human traffic between those locations?
I don't know. I mean, I don't think so. We think that, so all of these samples were just collected in the Boston area from one house per town. So it could be something like that. It seems like there's a lot, like significant inbreeding within one house, and that there's not a lot of gene flow in between those, and that we're not really spreading a ton of moths back and forth as we go from house to house. But in the future, hopefully we'll have more information about that. Yeah. Cool. Thank you. Okay, I think if there's no more questions, then that's all. Thank you so much.
Hello everyone, my name is Natalie Van Dis, and today I'm very excited to talk to you about the winter moth's genetic adaptation to climate change. So this picture tells you the story what happened to the winter moths. So this species, they overwinter as eggs, and their egg development rate is highly temperature dependent, so that in the past, which you see on the left side of this picture, climate change was causing these eggs to really hatch more than 10 days before their food source occurred, which is oak budburst. And under this immense selection pressure, they were able to rapidly adapt so that nowadays, which you see on the right of this picture, they hatch around the timing of oak bud bursts. So this example of genetic adaptation to climate change is still quite rare. I just finished this systematic literature review, and in total I found 40 examples of species of 9 different taxa that are showing this genetic change over time in response to climate change. And if we look at these examples taken together, they generally still have very low temporal and spatial replication. So usually they only sample 2 time points for 1 or 2 populations. Also, there are still very few studies that have gathered genomic data for these examples, and in fact, there are currently no genomic studies that have more than 2 time points for more than 2 populations. Well, I think the field could really capitalize on these examples to answer important questions about adaptation to climate change. So we were really keen on doing that, and we wanted to use high-resolution temporal genomics to gain insight into the drivers of rapid adaptation to climate change, capitalizing on this example of the winter moth. So we were very lucky with the winter moth to have nice long-term data available for 4 different populations, which you see here on the map. They're all in the Netherlands. And there's this yearly sampling been going on since 1994 where we collect winter moths, but we then also measure the selection strength in the field, so the amount of mistiming between this winter moth egg hatching and the oak bud burst. And from that data, we know that the winter moth has been adapting to climate change. So in this figure, the 0 line is the timing of oak bud burst in green, and then in black you see the timing of winter moth egg hatching relative to that. So you can see that in the 90s they were really mistimed, and over time this has been decreasing. And from repeated experimentation, we know that this has been due to a genetic adaptation. So there have been genetic changes in how a winter moth egg development responds to temperature. And we were really keen to dive more into that using whole genome sequencing, so seeing if we can find the actual genomic regions that have significantly changed over time. So we did DNA extractions, choosing 4 to 11 time points per forest, where each forest was showing a similar phenotypic trend over time. And then for each forest, we had this starting year, which was the year 2000, and the end year 2020. And then depending on how many samples were available, we chose multiple time points in between as well. So that involved doing a lot of DNA extractions, which we then pulled together so that for each time point and each forest, we have these pools of 30 individuals, where we have a 50-50 sex ratio, and we then used Illumina sequencing to a depth of 60x. So the first interesting result already came at the variant calling stage, where we found really enormous amount of genetic variation. So this is the raw output of our variant caller, and if we just look at the SNPs, we found more than 41 million SNPs in these quite local populations. So we think this indicates that these populations seem to have very high levels of genetic variation, which is of course an important driver if you need to rapidly adapt. For the analysis, we couldn't look at all of these variants, of course, so we, for now, decided to look only at biallelic SNPs and then did some quality filtering so that we ended up with 3.5 million SNPs per forest for the eventual analysis. The first thing we wanted to know was to see if there's actually any population genetic structure, so are these forests really separate populations? And we used the fixation index for that. The way we did this is that we both looked at the FST values in the start year and then also in the end year, so that if these pairwise comparisons would fall on the diagonal line, then there's no change in population structure over time. So from the axes of this figure, you can already see that the FST values were very low, and combined with this very high genetic variation, we think this indicates that these populations must have a very large effective population size, which of course makes selection more effective, so could be a driver of this rapid adaptation. So this is what the actual pairwise FSTs looked like. So even though the values are low, they were significantly different from 0, and they were not deviant from this diagonal line. So there is some population genetic structure, and it has remained stable over time. So then the next thing was really trying to find these genomic signatures of selection, and we really wanted to make full use of our temporal design by looking at these allele frequency trajectories. So here's an example of one SNP that is showing this type of pattern that we might expect from this continued climate change selection, where this is gradual increase in its frequency over time. And we looked for these type of SNPs by running a simple linear model. And then we compared those results to our null hypothesis, which was running the same model again for every SNP, but then shuffling around the time labels. So basically asking, how often do you see a pattern like this just by chance? So these are the results from that analysis. So here we have a Manhattan plot, one panel for each of the forests that we sampled. And then the pink diamonds are the SNPs that meet our significance threshold. So these are SNPs that show a significant change over time. And you see that for each forest, there are some of these pink SNPs, and they're spread out across the genome. And when SNPs are like marked in red, they were significant in more than one forest. So you can see that there are not that many of them. And actually, when we not just look at the significant SNPs, but really look at the top 10,000 SNPs for each forest, we really see very little overlap between these forests at the SNP level. So that's an interesting result. But in general, I think at the SNP level, there's not that much signal going on. But of course, we did pooled sequencing, so we also wanted to increase our power by doing a window analysis. And that window analysis is now also plotted on here. So we did 100 kb windows, and these orange diamonds at the bottom of each panel are significant windows. So these are genomic regions that have significantly changed over time. And you can see that now we're picking up a lot more signal for most of the forests. But again, if we compare them, so to see if there's any overlap, looking at the top 100 windows, we again really see very little overlap. So this seems to indicate that despite these 4 forests having a similar phenotypic trend over time, there seem to be multiple ways to roam. So different genetic underpinnings to get to that same phenotypic change. We were keen on looking more into the trait architecture for that. So we used RNA-seq to see if we could find candidate genes that are responsible for regulating how this winter moth egg development rate responds to temperature. So we gave embryos at different stages in their development a change in temperature, and then looked for genes that change their expression. And I can't go into depth into this study for sake of time, but the take-home message that we found a huge number of temperature-responsive genes. So more than 800 genes significantly changed their expression in response to a change in temperature. So we think this indicates that this trait has a polygenic architecture, which corroborates those multiple ways to roam, which could, of course, increase the likelihood of adaptation and might be a driver of it. So now we have both whole genome sequencing data and also RNA-seq data. So we were super keen on overlapping the two and seeing which of these candidate genes actually are showing genetic change over time. So I've added that now to this Manhattan plot. So in each panel at the top, you see these black squares, which are candidate genes that overlap with our significant windows. And if we look across the 4 forests, we found that 55 of these temperature-responsive genes are showing genetic change over time. So these are really putative targets of selection that we're really keen on investigating further. And I had a quick look at them, and some of the genes that popped out are cell cycle regulators, genes involved in transcriptional control, and also genes that are involved in environmental resistance. So to summarize, we use temporal genomics to find drivers of rapid adaptation to climate change using the winter moth as our system. And based on our results, we found we would propose 4 drivers. The first one being this very high standing genetic variation that these populations are showing. The second one, that they seem to have a very big effective population size. Then the trait that has been under selection has a polytrenic trait architecture, so these multiple ways to roam could increase the likelihood of adaptation. And finally, the fourth one, which I couldn't talk about in the main talk, but I still wanted to mention, is that we found hard selection in the winter moth. So we used population modeling to show that the climate change selection has been so strong that it has actually impacted their growth rate, which we think could be an important driver of rapid adaptation. So I have some time left, so I wanted to also talk to you about what I plan to do next, because I'm definitely not done analyzing this really exciting data set. And the next thing we want to do is look more into the eco-evolutionary dynamics of this species. Because we not only have this temporal genomic data set now, but we also have ecological long-term data, which I now plotted here at the bottom. So in black, you see for every year that we sampled the population size, and in red, we see the selection pressure, so the mistiming that these winter moth experience. So it would be super cool if we can combine the two and see if changes from year to year, the magnitude of that, or just the slope of the change, so how fast it's going, is that somehow influenced by population dynamics and selection strength. And I'm not only working on that in the winter moth at the moment, but I'm also doing a postdoc and having a similar data set in a different species. And I'll have a poster about that tomorrow. So if you have any ideas on how to analyze this temporal genomics data and combine it with ecological data, I would be super keen to talk to you. So please also come and visit my poster tomorrow. So with that, I want to thank all the people that were involved, including co-authors, my supervisors, and also our funders. And I want to thank you very much for listening, and I'd be happy to take some questions. Any questions? Yes?
Maybe I missed it. Could you just explain how which variables to assess the evolution of the non-natural?
So I assessed all alleles. So those 3.5 million SNPs that I had after filtering, I did this linear model for all of them. And that's what you saw in those Manhattan plots. You're welcome. Any other questions? Yes?
Did you have any outlier loci on the Z chromosome, the sex chromosome?
I don't know by heart, but maybe I can quickly pull it up, because we still have some time. Yeah, there are definitely some outliers on the Z chromosome. Yes. Thank you.
Can everyone hear me? Okay, thank you. So I'm really excited to tell you a little bit about our work on pathogen evolution today. So pathogens face many different selective pressures in their environment. These include predators, the hosts that they infect, the other microbes that are in their host, as well as having their own parasites. In addition to having these biotic selective pressures, pathogens also face abiotic challenges like antimicrobials, resource limitations, and extreme temperatures. And like other organisms, it's unlikely that pathogens are facing these selective pressures one at a time. And so our big question today is, how do multiple selective pressures shape pathogen evolution? And we're going to focus on antimicrobials and hosts. One type of antimicrobials is antibiotics. The first clinical use of antibiotics was in 1940, and in just a few decades, the misuse of antibiotics have led to the global crisis of antibiotic resistance. We know that exposure to high concentrations of antibiotics can lead to selection for high resistance in pathogens, but exposure to low concentrations of antibiotics could also shape pathogen evolution. So one way that we can determine how susceptible a pathogen is to an antibiotic is by determining its minimum inhibitory concentration, or the MIC. This is the lowest concentration of an antibiotic that can prevent the growth of the bacteria. So sub-MIC is the concentration that is lower than the MIC, and it allows for susceptible bacteria to grow. Sub-MIC levels are actually found in a lot of different environments, including in water and soil, from waste and misuse. And sub-MICs also exist in the environment naturally from microorganisms that produce antibiotics. Variations in antibiotic concentrations can also be found in treated individuals like humans and livestock. And we know from experimental evolution studies that sub-MICs can have strong effect on pathogen evolution despite exerting weaker selection. But a lot of these studies have been done in vitro, and we don't have a very good understanding of what happens when there is a host involved. And so separate from antibiotic selection, we know that hosts alone can exert very strong selection on pathogens. Hosts can have different forms of defenses, like the innate and adaptive immune systems, as well as having defensive symbionts that can protect them from pathogen infection. Another sort of selective pressure associated with hosts could be nutrients, which can vary across sites within a host individual, as well as the nutrients inside of the host can be different than the nutrients outside of the host. And we know that host factors alone can affect pathogen traits like virulence and antibiotic resistance. We know that sub-MICs alone can affect how pathogens evolve. So what happens when these two selective pressures interact with one another? So that's the question that we're trying to address with our study. Our prediction is that when both the host and sub-MIC antibiotics are acting simultaneously, that there would be a trade-off between virulence and antibiotic resistance because the pathogen response to selection is impeded. Or alternatively, these two selective pressures could actually interact synergistically and facilitate pathogen adaptation. And the two traits we're interested in are virulence and antibiotic resistance and how they evolve in response to one or both selective pressures. And so to test our prediction, we evolved a pathogen in different environments. The pathogen that we evolved is Staphylococcus aureus. It's an opportunistic pathogen that can infect a wide range of hosts. To start out our evolution experiment, we had two ancestors. The first ancestor is methicillin-resistant Staph aureus, or MRSA. MRSA nowadays actually refers to resistance to multiple drugs, not just methicillin. Methicillin actually isn't being produced anymore. It's been largely replaced by other drugs, including oxacillin, which is what we use for our study. The other ancestor in our study is methicillin-sensitive Staph aureus, and these two ancestors are isogenic, except at the locus that confers antibiotic resistance. To put this into context, it takes 16 micrograms per mil of oxacillin to prevent the growth of the methicillin-resistant staph, and only 0.25 micrograms per mil of oxacillin for the methicillin-sensitive staph. And so we took a clone from each of these ancestors, grew them up in broth culture, and then divided them into replicate populations that we passaged independently. Half of them we passaged in the host, and then the other half in the absence of a host. The host that we use was C. elegans nematodes. C. elegans has been used as a model system to study virulence factors in staph. We divided these populations up even further and evolved half of them in the presence of sub-MIC oxacillin and the other half without oxacillin. And so our overall experimental framework is that we have 8 treatments with 6 replicate populations each that we passaged 12 times. We have methicillin-resistant staph that were passaged in the presence or absence of sub-MIC oxacillin with or without a host, and the same for the methicillin-sensitive group. And so this is the labels in the purple and yellow boxes are what I'm going to use to label the treatments in our results. They're essentially the environments that the pathogens evolved in. And so first, we looked at virulence evolution. We took the evolved populations and we used them to infect nematodes, and then quantified host mortality as the metric for virulence. So on the x-axis, we have the experimental evolution treatment. On the y, host mortality, and then the dotted and dashed lines indicate the virulence for the respective ancestors, and we can see that they have comparable virulence. So for the methicillin-resistant pathogen, we saw that populations that evolved under selection from both the host and sub-MIC antibiotics selected for the highest level of virulence. So having pre-existing resistance to antibiotics did not impede pathogen evolution. We saw a similar pattern with the methicillin-sensitive group, where, again, both selected pressures selected for the highest level of virulence. So both of these results suggest that regardless of the ancestral antibiotic resistance status, virulence can increase. And so in terms of virulence evolution, exposure to both selective pressures did not hamper pathogen evolution. And Next, we measured antibiotic resistance evolution. We took our evolved pathogen populations and we determined whether they would grow across a range of oxacillin concentrations. Here, the y-axis is showing the number of populations with a particular MIC. The range of oxacillin concentrations that we used was in between the MIC for the methicillin-sensitive ancestor and the methicillin-resistant ancestor. So for the methicillin-resistant group, we saw that antibiotic resistance is mostly maintained across most populations, except for 2 populations that evolved in the absence of oxacillin, which isn't too surprising because resistance could be costly. But I think what's more interesting is the methicillin-sensitive group, where populations that evolved under both selective pressures had greater MICs than the ancestral MIC, up to 8 times the concentration that would have killed the ancestor. So here we see that exposure to both selective pressures actually decreased drug susceptibility. So in addition to phenotypic evolution, we also looked at genomic evolution. So we took these populations and conducted whole genome sequencing on whole populations. This is a summary graph of the mutations that had arisen in both ancestors. It's a lot to dissect, but one pattern that we noticed was that a lot of these mutations occurred in genes that regulate other genes, suggesting that there's potential for pleiotropy in pathogen adaptation. One gene that we are interested in particular is this gene called COD-Y. It's one of these regulatory genes, and we're interested in it because it has the potential to link antibiotic resistance and virulence. So why do we think this? One bacterial trait that can mediate both antibiotic resistance and virulence is biofilm production. Biofilm is this aggregate of cells that can stick to surfaces. So we know that in staph, biofilm reduces efficacy of antibiotic treatment, and it's also involved in staph pathogenesis. We know that sub-MIC level of antibiotics can induce robust biofilm formation, and biofilm can enhance virulence and protect staph from host defenses in C. elegans. And it turns out that no mutants of COD-Y can induce robust biofilms. So we went and measured the biofilm production of our populations. So for the methicillin-resistant populations, we saw no differences in the level of biofilm production across treatments. For the methicillin-sensitive group, however, we saw that populations that evolved under selection from both the host and oxacillin increased biofilm production. And from our genomic data, this was the treatment that had the majority of the COD-Y mutations. But we also saw that in the absence of a host, exposure to oxacillin could also increase biofilm production. And this treatment had no COD-Y mutations. And so one thing that could be happening is that since biofilm itself is controlled by multiple genes, the increase in biofilm production in both of these treatments could be due to mutations in different genes. And so here, we saw that selection from both the host and sub-MIC antibiotics increased biofilm production, and that this evolved trait has the potential to be due to pleiotropy that can confer both decreased drug susceptibility and increased virulence. So to go back to our question in the beginning, how does selection exerted both the host and antibiotics affect pathogen evolution? We saw that they can actually interact synergistically with one another and facilitate pathogen adaptation. And I think the overall message here is that environmental changes due to human actions, like adding antibiotics into the environment, can have the potential to shape how pathogens evolve. And when we add a host into the mix, this can lead to potentially negative consequences for the hosts. So with that, I would like to thank everyone here, and thank you for listening. Yeah, I think that's a good hypothesis. I know genetically they've lost, what confers their resistance is a mobile genetic element that encodes for a gene that allows them to be resistant, and those two populations have lost this like locus that confers resistance. And so I think it could be interactions with the host, and also it could be I guess having this mobile genetic element could be costly to them as well. Thank you.